everyone and welcome back to my channel Be Yourself. This is Dr. Rajni Sharma and today I'm going to discuss some of the important topics for the CSIR JRF net. Actually, I am releasing this video just before the examination because in this last week, students generally get confused with which topic they should revise first for their examinations. Though I can't say this topic will definitely come in the examinations or not, but these are the expected topics for the CSIR JRF net. So let's start. I will discuss unit wise the topics. So let's see which topics are important. So here is the syllabus for CSIR JRF net. I have downloaded it from the website of CSIR. These are the 13 units of the CSIR JRF net, and I will discuss the unit wise. First is the molecules and the interaction relevant to biology. In this, mostly questions are asked from the pH and buffer and the chemical kinetics or the reaction kinetics like the K1, K2 values. pH, it can be asked like pH numerical based questions are asked basically from the pH and buffers, and in thermodynamics basically Gibbs free energies and which is also we term it as del G is asked in the numerical based questions from this topic. Next is the bioenergetics and glycolysis, the section E, where the students must remember or mug up the number of ATP or assimilatory powers released during the bioenergetics or glycolysis, like how many of ATP are synthesized at the end of glycolysis and what are the steps where the ATP is synthesized or used and where the NADP is produced and how they are linked with other cycle, like how pyruvate at the end product of glycolysis enter into the Krebs cycle and how they are processed. So this must be very clear to the students in this last week. Better revise all these topics. Next come the enzymes. It is number F, enzymes and enzyme kinetics. I have a video over this enzyme and enzyme kinetics where basically numerical based questions based on the turnover number and specificity of the enzymes are basically asked. I have a video over this, how to calculate the turnover number or the enzyme kinetics, in my channel Be Yourself. At the I have given the link of my channel in the descriptions, and in the end I will show you how to see the description and how to get the link. So let's continue along with this. And next is that Ramachandran plot is always asked during the examinations. It can be in section B or can also be in section B. Last time in total 8 marks question was asked from Ramachandran plot. So we must have a clear-cut concept about Ramachandran plot. What is the value of psi and phi for alpha helix, beta pleated sheet, and for example, if psi and phi will lie negative, for both will be negative, then it will be right-handed alpha helix. Likewise, such types of concept must be clear about the Ramachandran plot. And we know that in Ramachandran plot we can recognize the alpha helical form as well as beta pleated sheet in this, and also we can study the collagen triple helix in this Ramachandran plot. And about the conformational nucleotides acid, basically tRNA and microRNA, or we can also known as SNO-RNA or siRNA. So you should revise these topics first. And about the metabolism of carbohydrates, lipids, amino acids and nucleotides and vitamins, we in these steps also during the metabolisms, how many ATP is released, how many consumed, and what are the regulatory pathway for the metabolism, carbohydrates, lipids and amino acids, nucleotides must be clear. And in the last from the last year's questions, what I analyzed, simple pathway was given there. Only you have to identify which enzymes should be used in that pathway. So enzymes should also, you should always remember the pathway and along with the enzymes and how they are oxidizing or reducing a particular compound in a particular compound. So come to the next, which is the cellular organizations, and here the membrane structure and functions basically asked from the plasma membrane active transport, how the ion channels are involved, what are the membrane potential. What I will prefer a book Cooper for the cell biology, and I will basically suggest you to only go through the figures of that book, and you just read out just a caption of that figures, because that book is very much specific for the topic, and you will, even if you will go through the figures only and also only you will read the caption, you will able to cover whole of the topic related with the cell biology. So better revise from the Cooper. If you have other book also and you felt that book easy for the revision, then you can also prefer, but I basically prefer that Cooper for the revision, last time revision only. The figures and caption you should go through that. I have few videos also on cell biology in my channel. I will give the link at the description, you can get it from there. And now where is the cell cycle and cell division, especially CDC, and how MCM complex are involved during the replications, and how different types of CDK and cyclins are involved from the passage of, or the to clear the checkout points, like stop points, or you can say the to clear the one stage to another, from G1 to G2, G1 to S to G2, and from M. What are the cytokinin and CDK are involved during the cycle? Remember that point. And finally, the microbial physiology. It is growth kinetics are always asked from the past. Whatever I have analyzed from the past year questions, that it was definitely there. And the strategies of cell divisions that also include the same CDK and cyclins involved, and how they get degraded, what the function of V, what the function of definite cyclin. So for example, cyclin B is involved for the passage of G2 to M phase. So these types of things you should remember in your mind in this last week. Next unit is fundamental processes, which include the DNA replication, repair, or recombination, or you can say the central dogma of our cell, which is replication, transcription, and translation. Remember once from you can better go through your notes, or you can also follow some videos on YouTube, and if you are having some notes, then better revise through that, how what are the units of replications, and which enzymes is basically involved in replication, what is the direction of replication, and which enzymes is involved in the repair, recombinations, and different types of repair, where why this particular types of repair is there. This point should be clear in your mind. And also in RNA synthesis, basically as I have gone through the past year questions, then I come to know that different types of RNA polymerase in eukaryotes 1 to 3 and is there, and which polymerase synthesizes which RNA is basically asked. This is basically asked in the section B questions, but some twisted forms of questions are generally asked from the fundamental processes in the section C. It can be if you will talk about the RNA editing, then you must know about the guide RNA, siRNA, miRNA, how they are involved in the RNA editing, and trans-splicing. Along with simply splicing of introns, you should also revise once trans-splicing process, how it occurs, and what is their phenomenon, how it is involved, what is actually involved in that process. You better go through it. And in protein synthesis and processing, amino acyl T transferase activity is basically asked. Don't forget to revise this topic also. Apart from this, other questions can also be asked simply based on the genetic codons, that which codons code for which, and what will be the anti-codon for the particular amino acid on the tRNA. As we know that messenger RNA will be coded, where the amino acid which involved in the protein synthesis will be recognized by a specific type of tRNA, and there is certain energetics involved for the specificity or the high fidelity of the RNA synthesis. So you must revise once that also. And there is a control of gene expression. This is very important topic, and in this is also a easiest topic because questions are not asked on the memory basis or how or what you know about the topic. Questions are generally asked as some twisted form, or some paragraph can be given, and then you have to analyze that paragraph and or the graph given in the paragraph in the questions. Basically graphical questions or analytical questions are asked from this section. Next is the cell communication, cell signaling also, in as there are certain links are there. Basically pathogen induced diseases, you must mug up all the pathogens involved in the pathogen based diseases for the animals and plants including human beings, because we are including animals. So you must mug up all the pathogens and their transmittance. I will give a link in the description. I have some collection of pathogens induced diseases in animals in tabular form. I will give that link in my description. You can get it from there. And another cell signaling, how G couple proteins is there and how IP3, IP2 is involved, especially glucagon and glucose metabolism, how receptor based cell signaling are involved during the communications of the cell. So these are the basic topics involved asked during the examinations. So every topics and involvement, you can't say that no, this time this topic is not going to be included in this examination, but these things you must revise at the end of the end week of your examinations. And in number C section, cell communications, about the regulation of hematopoiesis and general principles of the cell communications and integrins and all. You can better for this also, for the gap junctions and extracellular matrices, these questions are definitely asked each and every time. So don't miss this topic, especially the gap junctions, extracellular matrices and integrins. And for this also I will refer Cooper, go through their figures and captions only. And finally cancers, basically what are the factors involved in the cancer, how they spread, are basically asked. And this time, as it was asked that which types of cancer can be there, some basic questions can also be asked. And what are the factors like viruses, what are the inducing factors for the cancer, you must revise once that factors also. And apoptosis is definitely asked in the examination, so don't miss apoptosis. It is must asked in this examination also. This time also it will be definitely asked. And in the immunity, don't forget to revise about the toll-free receptors and about the antigen antibody interactions. What are the like ELISA test and all, precipitations, how the precipitations will take place, why it takes place. Basically ELISA or some technical based questions, or we can say techniques based on the immunoprecipitations are generally asked based on the immunity. Or some autoimmune diseases are there, you must mug up all these. Hypersensitivity is also there. These topics are basically very important. And once go through the viral infection, HIV infections, how it infects our body and how it regulates its own synthesis and infect our body. Revise that. Developmental biology, I think developmental biology you can score very much positive marks in developmental biology because it is generally simply based on the theoretical questions. There is no concept, no techniques to understand these questions. Basically once go through the different certain terminology related with the developmental biology, like commitence, specification, induction, competence. Apart from there are certain other basic terms also, like delamination, involution, ingressions. Once go through this simple terminologies to score high in developmental biology. And very high percentage of the questions are asked from the developmental biology, so don't miss this unit. And apart from this, the controlling factors involved in the fusion of gametes and access and pattern formations is there in section C. Access and pattern formations in Drosophila, amphibians and chicks are there. In organogenesis, once go through the vulva formations in C. elegans, eye lens inductions, limb development, and in this also it is indirectly related with the limb development. It is indirectly related with the apoptosis. So this can be linked with the apoptosis, or the question based on the limb development and apoptosis can be asked in integrated form. Sex determinations is also very important topic. So these are the some important topics we must revise before our examinations. And the morphogenesis and organogenesis in plants, basically it is asked in the or ABC modeling of flowering is very much important in developmental biology. It is also included in the physiology of plants. But ABC modeling, which determines the flowering, how flowers are synthesized. Actually, there are three genes A, B, and C, which regulate the flowering process in plants. So once go through that, you can get it from book Gilbert. I preferred Gilbert for developmental biology. You can get this ABC pattern of ABC model of flowering in from Gilbert also. Again, programmed cell death or apoptosis is there, aging and senescence. You can follow same book Gilbert for this. Next unit is the system physiology of plant. Go through the C3, C4 and 6 CM pathways, mug up it completely and how these are interlinked, and what is the difference between C3 and C4, the chances that it will come this time also. And respiration and photorespiration, same. What are the types of how many ATP or NADPH involved during this process, or the assimilative power involved there, and how the other and the regulatory sites for these cycles. Must keep that in your mind. In nitrogen metabolisms, remember the bacteria involved during the nitrate or ammonium assimilation, and also once go through the chart. I will prefer these for plant physiology. You can there are certain charts and the cycles are there in that book. Remember all those things. Once have a glance over this, because their options are there. So if you will, even if you once you will go through the chart and cycles, then that point will be in your mind and you can easily attempt. And because there is no twist in these questions from the plant physiology as well as from animal physiology, they are direct questions, so you can easily attempt such questions and you will not score negative in these questions. And another is the plant hormones, also in from Thais book from book by Thais for plant physiology. You can simply revise the pathway or the biosynthesis pathway for different enzymes, 5 types of enzymes in plant hormones, and what are their particular role. For example, as ethylene, what is the particular role of ethylene, abscisic acid. So you must be remember that points. Next is the sensory photobiology. Sensory photobiology, basically I will prefer don't miss phytochromes. Phytochromes or there are two types of phytochromes. Basically they are the far red and also known as PFR or PR. Different types of genes involved in phytochromes, so don't miss that topic. Biological clocks are always asked, and from the previous year question I also analyzed that these are repeating from last few years. So once revise the biological clock, and this is the very simplest topic. Solute transparent and photo assimilation translocation. In this there may be chances that numerical based questions on the water potential can be asked. So once go through their formula and how transpirations regulate the physiology of plants. Such types of questions can be asked. And the biosynthesis, for the biosynthesis of terpenes, phenol and nitrogen, there is a very good chart in this in plant physiology. You prefer that book and just once go through that chart, you will able to solve any questions from based on the biosynthesis of terpenes, phenols and nitrogenous compound and their roles. Stress physiology, you just once go through the stress physiology, just swipe your pages of this chapter, because this is the easy one and you can easily keep all those things in your mind. Next unit is the system physiology of animal. In this you must remember the blood compositions, serum and plasma, what is the difference between serum and plasma, and don't miss ECG, because from the last year questions every time it is repeating ECG. This time it was in last year this was in 4 marks questions, so it can be either in the B section or C section, but it will definitely be there, I think so. Next come the respiratory systems, how there is exchange of gases is there, what is the role of hemoglobin for the exchange of gases, once go through this. In the nervous systems, once go through the action potential of the neurons, and how acetylcholine and calcium ion involved during the transmission of nerve impulses from axons to neurons, dendrites. So you once go through this. Next come the excretory system. In excretory systems, remember the regulations of water absorptions by the involvement of certain hormones like oxytocin and vasopressin. Once go through this. About the thermoregulations, how how body get acclimatized to the adverse conditions. Some it can be asked in this time also. Next, BMR, must go through the simple definition of BMR, basic metabolic rate, and a formula involved with the BMR. And again, next topic is inheritance biology, based on the genetics. Basically, once you can revise it from any of your book or any notes, what the dominance is there, what the independent assortment is there. We always, these things are always in our mind, but some types of tricky questions are asked based on the Mendelian principles, and basically based on the ratios of epistasis, or the and what are the basic principles involved in the expressions of a particular genes, like what the dominance epistasis is there, and expressions of what the dominant or recessive genes will express the particular trait or not. So these things you must have in your mind. Dominance, codominance, phenotypes, sex linkages, and basically the mitochondrial and chloroplast genes. Once go through the difference between the basic difference between the mitochondrial and chloroplast genes, and what they about the transformation, conjugation, transduction, and sexduction, and the maps interrupting the mating. One question can be asked from the conjugation based on their transmission from the F positive to F minus. Based on it can be a concept based questions, that if there are 4 amino acids there and the different times required during the conjugations, based on the timing of their conjugation, the sequence of amino acid we can determine. So such types of questions can be asked. In human genetics, pedigree will be definitely be there. One question will definitely be there from the pedigree analysis. Polygenic inheritance is also important one. In mutations, simple questions are asked from the mutation. You can easily attempt that. Then how mutations can take place, frame shift mutations, what are the chemicals involved in the frame shift mutations, and how, like example, how acrodyl is involved in the mutations, how basically we can say that what are the mutagens involved. We must have that in your mind. And some sequence based questions can also be asked from the mutations, that whether it's deletions or duplications, or some concept based questions can be asked from the mutations. In recombinations, generally site specific recombinations are asked, and role of RAC-A, what the role of RAC-A, such types of questions can be asked. Move to the next unit, which is diversity of life form. The principles and methods of taxonomy. I will prefer you NCERT books to revise the classifications of both plants and animals, because questions are definitely asked from this units, and I will basically prefer you +2 NCERT books to revise this chapter. And ecological principles are, and case selection is very important, or the survivorship graph is very important for the in the ecology portions or the in 10th unit. And you must have some terminology in your mind, like what the damage means, what the niche means. Simple terminology, you can keep that in mind, and this is a direct question, there is no twist. You can easily attempt question from ecological portions or 10th unit. And this is the scoring, never miss this one. About the next what will be important, almost every topic is important in this section in unit number 10. Basically R&K selections are asked, and any more questions, let me see once have a glance over this. ICS island biography can be asked. Applied ecology, like biodiversity status monitoring and the measure biodiversity changes can be asked. Yes, one have take out the list from the net, that what are the different project, tiger project, biosphere reserve project is there. Once read out it, because it just matter to read out, nothing to mug up in this. You just go through this topic. Yeah, this will be in your mind in this last week, so no need to panic about this. In evolution is also very scoring unit. Just all go through all these topics, just once glance over this, like operand and Haldane, concept of operand Haldane lemmas, Darwin concept of variations, first cell, evolution of eukaryotes. These are also interesting, and if you will once go through the chapters, you will this will be easily in your mind. And the molecular evolutions, how molecular the concept of neutral evolutions, and the molecular clocks. These are the important topics. For gene duplication and divergence, these are the important topics. The mechanisms, yes, gene pool, gene frequency, Hardy-Weinberg. One question must be there from the Hardy-Weinberg law, so don't miss the Hardy-Weinberg law. It's quite simple, and this is just based on the formula, random genetic drift. Very interesting topics, and also very easy to attempt, and it will never score negative marking, especially the numerical based questions. What is the allopatricity and sympatricity, must have these definitions in your mind, and how the graph varies in the selections process, about the natural selections, how the direction what will be the variation in the graph if it will be directional. So this thing, once go through it. Another is skin selections. Another topic is skin selection is also very important, and altruism is also important. Now come to the next topic. Yes, I am forgetting this parental care. Once go through this parental care, because many of times these questions are asked from the previous question. What I have analyzed from the past year questions, based on the migration, orientation, and navigation. Next unit is the applied biology. In applied biology, microbial fermentations can be asked. Application based on the principle and vaccination process, and their diagnosis process can be asked. In diagnosis, some diagnostic tools can be involved in the questions, like what will be the diagnostic tools for the particular diseases, and how we can tag it with some types of fluorescent compound or protein, or there any reactant for the diagnosis of particular diseases, or simply we can say that western blotting, eastern blotting can be asked in this applied biology section. Molecular approaches for diagnosis, like hybridization techniques and all, can be asked. Strain identifications, like which strains of bacteria or virus bacteria can be there. IMVIC test we can use for the strain identifications. Once go through the IMVIC test for the strain identifications, and what was the specific dyes to identify about spore formations, spore forming bacteria, gram positive, gram negative. Very simple questions can be asked from this. And molecular markers can be asked, like RAPD, FLP. These questions will definitely be asked from this. Biosensor, what is the biosensor? So these questions are very important. And last in the methods in biology or biology is there are three section, or no, sorry, there are 5 sections. No, there are many more sections in this. So isolation purification of DNA, I can be asked as how we can isolate the RNA. cDNA can be asked, or the two-dimensional gel electrophoresis. There basically analytical questions are asked from the methods in biology. Some gel based questions will be there, that different band will be there, and you will have to identify the diseased band, or we have to relate with the son with his father, who is the carrier one. Such types of question can be asked in methodology in biology, methods in biology. And the DNA fragments, difference between the DNA fragments and prokaryotes, prokaryotic and eukaryotic DNA, you must have once go through this, so that some concept based question if you will be asked, you can easily attempt. Yes, different cDNA libraries in plasmid, phase, cosmid, plasmids are there. Different types of plasmid. What are the conditions we should keep in mind for designing a plasmid? How we can use the knockout genes to study the function of particular gene in bacteria and other eukaryotes also. And other post-translation modification in the protein, how it will change the function of the protein and how it will be induce the receptor or any particular marker on a protein, so that it will go through a definite trafficking pathway or the site for their action. So such questions can be asked. Isolation, separation, analysis of carbohydrates and lipid molecules. We can say the chromatography or HPLC questions can be asked based on the HPLC chromatography. And histochemical and immunotechniques, ELISA I already have discussed that ELISA question based on the ELISA Western blot can be asked. You must have a clear-cut concept about this or the principle and its applications. So because questions are not asked directly from such technical question, from such techniques, there will be twisted, or some experimental based questions can be there, some experiment will be there, and then you will have to analyze the result for that. And next is biophysics method. For biophysical method, go through all the principles for UVR, fluorescence, and all the related topics and their applications, and what are the special conditions are used. For example, NMR are used to analyze the larger molecules. So such types of concept must be there to attempt such questions. And in statistical method, generally chi-square tests are asked, and from the previous year questions what analyzed these times, the simple theoretical questions are asked, and why we are not using jet test, rather we are using t-test. T-test is used for the small sample size and jet for the large sample size. These types of questions can be asked. And what will be the value of mean, or what for the acceptance of particular theory, what is the theory, hypothesis, how we take the hypothesis, and what are the conditions to accept the hypothesis, and why will not reject the hypothesis or accept the hypothesis, what is the p-value. Have these things in your mind. Radio labeling techniques, basically radioisotopes decays are generally asked, and for detection process used for the particular disease are generally asked in the question. Freeze etching technique and freeze fracture methods for EM is very important. Patch clamp recording is what is patch clamp recording. ECG I have already discussed. Once go through the principle and applications of this PET MRI and fMRI and Kcat. And in the methods in biology and field of biology, like sampling method, what are the sampling methods for your biostatical analysis? Is there behavior, or the what is the remote sensing methods? This is very important one. So these are the topics which you can revise at the end at this last week of your examination for CSIR JRF net, so that you will not miss the questions from this. So all this was in this video. In my upcoming videos, I will try to make a short videos on how to score positive marks, how to avoid the negative marking. So don't forget to subscribe and share my channels. And one thing I have forgotten, I have told that I will told you how to get link from the descriptions. Here I am opening my Cyber, and when you will go in YouTube channel, take any video from YouTube, basically I will opening my own apoptosis by be yourself, sorry, by be yourself. As this is my video on the apoptosis, I better pause this video. And now when you will scroll down, when it was published, and there will be so more. This is the known as descriptions. I am giving in this video, you will get the link of my channel and also some notes based on the animal larva, especially from the classification of animals. You will get the link of some notes in my descriptions. So better after this video, better you scroll down and get the link from this descriptions. So this is how we can go and see the descriptions. I have given the link of my channel and this video, as I unable to show this link because I haven't published at this video. I am going to publish, so I am showing you it from different of my videos. So you can get the link of my channel from this video in the description of this video. So this was in my video. Don't forget to subscribe and share my channel for more upcoming videos.